Coming to our last concept, Warburg effect. Warburg effect is very simple. You read this, you'll understand. Look at the figure, you'll understand. As you see, look at the figure. Look at the figure there. Differentiated tissue, well-developed tissue, differentiated, well-differentiated tissue. Compare that with proliferative tissue, highly proliferative tissues or tumors. See what is the difference there in terms of what will happen to glucose. Differentiated tissue, see, in the presence of oxygen, glucose is converted to pyruvate. That happens whether you have oxygen, doesn't have oxygen, it is a common thing. It will go all the way to pyruvate. And then the cell has mitochondria, sufficient oxygen, pyruvate is completely oxidized. And that will cause complete oxidation of glucose and it will give you 32 ATPs. Although we don't really focus too much on ATPs generation and that type of questions. But just for the accuracy, take 36 and add 32. That's the accurate number. 36 was an old number. Earlier, we used to consider every NADH as 3 ATP rather than 2.5 because that was a rounded number. 2.5, instead of writing that, just to not have confusion, you say 3. When I was student, I used to study it as 3 back 1996. Now it is 2.5, accurate number. Okay. With that accurate number, it becomes 32. So every glucose being oxidized to carbon dioxide, 32 ATP. Okay. So that's what is the result there. Now, even in the absence of oxygen and mitochondria, glucose converted to lactate and you get 2 ATPs, anaerobic glycolysis, 2 ATPs, 2 net ATPs. We know that now. Now look at proliferative tissue or tumor cells. In the proliferative tissue or tumor cell, whether your cell has oxygen or not, plus or minus oxygen, even in the presence of oxygen, and even in the presence of mitochondria, see what is the major fate of your glucose, what is the major end product of glucose oxidation in proliferative tissues and tumor cells? It is lactate. See, there is oxygen, there is mitochondria, but still 85% of your pyruvate is diverted to lactate. Despite having oxygen, despite having good mitochondria. The reason for this is this lactate further get into metabolic pathways and promote angiogenesis, proliferative effects, and all the things that is necessary for high proliferation of the cells is provided by this mechanism, like going into lactate, creating a little bit of acidic environment there, and then preventing apoptosis process. A lot of things in this angiogenesis process, proliferation process. And it helps in that aspect. So formation of lactate at high concentration by tumor cells will facilitate higher proliferation. It's a lot of concepts around it, not necessary at this point in time. Maybe in pathology 1, in that, carcinogenesis process, they might touch upon it. But the entire effect that you are seeing here, which is seen in the highly proliferative cells or tumor cells, is called as Warburg effect, or sometimes referred as Warburg phenomenon. Remember that. So whenever you come across this term, Warburg phenomenon or Warburg effect, this is what it is. Most cancer cells produce large amounts of lactate regardless of availability of oxygen and mitochondria. That is the statement. That's what you need to remember. Okay. So this is referred as Warburg effect.